All right, there it goes. Okay, so question about the lab.
Yes. So I was just wondering, I have them pulled up here. So the questions that we're answering, they're on the 13B. Is that right?
Do you
Both of
have
those?
do you want to pull it up on your
Yeah,
do you want to share your screen and then so we'll
sure.
know. And then students
Oh,
watch this recording will know too kind of what we're
it
talking.
says it's disabled.
Oh, let me see. So if I'm hosting, let's see. Oh, share screen, sorry. So multiple partners can share, event sharing, all participants. Okay, let's just go all participants. Okay, so now we can start sharing this one.
Okay.
Does it go? Did that work?
Yes, let me.
Okay. So share your screen and then I can comment on it.
Can you see that?
Yeah, perfect.
Okay. So I was just wondering if we just answer these questions.
Oh, so the one about your own ALU insertion.
Yeah, I was just
Yeah,
confused on what we're turning in, I guess, for this
yeah.
one.
So you're answering these questions, but like these are questions as if we had done a PCR and
Okay.
we would sequence yours and I'd run your sample on a gel. So go back up a little bit. So like this, so obviously this first one, what is your genotype for the ALU insertion? You know, you just make it up. And I don't know, I know I posted a video from last semester on this same stuff,
Mm-hmm.
but I don't know if I said that in that video. And then you're going to go through and you're going to score the heterozygous individuals, count them, how many homozygous, how many heterozygous, et cetera, figure out these frequencies. And then, yeah, that's what you'll hand in. More importantly for this is that you'll see a similar question like this on the exam, where I'll give you a picture of a gel and then ask you like these frequencies, is that population in Hardy-Weinberg equilibrium? So you want to make sure you really understand this lab because that's
Okay.
part of it. It's part of the molecular, what do we call it? Molecular genetics chapter. So this is one of the main kinds of problems. So you want to make sure that you understand how to get the genotypic frequencies, how to score the gel, how to get the genotypic frequencies from it, you know, or from the actual numbers, how to get the allele frequencies, how to convert those allele frequencies into the Hardy-Weinberg equilibrium, and then how to get an expected number of each of those genotypes where you can compare the expected and the observed and do a chi-square test and see if that, and be able to say, with sort of statistical certainty that the population is or is not in Hardy-Weinberg equilibrium.
Okay.
So yeah, so you want to make sure that, and this lab does a nice job of just kind of taking you through that step by step. It's first going to have you do it with a gel, then it's going to give you some data from, I think, the US population. You're going to do the same thing with that. You're going to have two chances that you actually test if the population is in Hardy-Weinberg equilibrium or not. So yeah, oh yeah, see, there's the US wide random population study.
So do we just do like a Word document and do 1, 2,
You can do that, or you can write, you can
or
do it
just
by
format?
hand and then just take a picture. You're saying to
Oh,
hand it in. Yeah,
yeah.
you just get it in any way that you can.
Okay.
Most people will write on this. Some people digitally write on these and then hand them in, but you can hand them in, yeah.
Mm,
You can make your own, yeah, Word file, or you can just write it in there.
okay.
Print it out or digitally write it in there and just do your calculations
Just-
right there. So either way is fine.
Okay. Cool. Thanks. And
Yeah.
so that will be a main point for this next exam. And then is this
Yeah,
one?
that's one of the main quantity, but you also need to, so if you go to that chapter, it shows you, how do you get in, it's just all about sort of Hardy-Weinberg stuff. So more, not necessarily incorporating the inbreeding coefficient F into it. The lecture does that, it shows you, okay, remember this inbreeding coefficient that we calculated a long time ago and did a bunch of problems with. Well, we can now incorporate this into the Hardy-Weinberg equilibrium and use it to look at some population genetic stuff. So that you're not going to do problems like that, but for instance, how do you see if a population is in Hardy-Weinberg equilibrium if you're looking at an X-linked gene, right? You look at the males first, you get the allele frequency from the males, then you can test it in the females. How do you do it with a recessive, condition? If you have a dominant recessive interaction between the alleles, and we looked at PKU in the lecture, and so you kind of have to be able to do that too.
Okay.
So those, and that's really the only quantitative stuff. The rest of it's just like about PCR reaction, the sequencing reaction, transcription, translation, all that's like sort of like just knowledge-based stuff.
Okay. And this is a dumb question, but is it all on new material?
Yeah, it's not. No, that's not. I get that question every semester,
Okay.
and in all of my classes. But in all of my classes, I don't do comprehensive finals.
Okay.
I think students are stressed enough at the end of the semester.
I know.
I hate to put more stress on them,
Yeah.
No, so it's just so
but then I was
transcription, translation, and the material comes from a bunch of different chapters. You don't have to, you don't have to know all of those chapters. We only talk about little, we sort of cherry picked some important parts of chapters just to get you exposed to this stuff at the end of the semester. So, for instance, microarrays, epigenetics. I told my face-to-face class yesterday, that for those of you who ended up doing that, watching that video, Ghost in Your Genes, it's great. We're not going to have stuff that's necessarily on there that's not talked about in the PowerPoint. So in the PowerPoint and the reading, more importantly, so remember there's that epigenetics reading. And so it's just some general stuff about epigenetics, general stuff about microarrays, and then the molecular biology or the population genetics type stuff.
Okay.
So the population genetics chapter is sort of the quantitative one, but it's really, the problems are just not going to be any harder than the lab. Might be a little bit different, some of them, like I said, with the X-linked stuff, with the recessive stuff, but it's not too involved. Not anything like the last 3 exams.
Okay.
All right. Any other questions?
I think that's all I have. So.
Okay, well, that's a short video too. I'll stop recording and then I'll wait a little bit to see if the one student shows up. But
Okay.
here, let me stop recording.